shovelnose rays, and intertidal sandflats. So first, I'd like to start off with an acknowledgement of the country. I acknowledge the traditional owners of the land where we meet today, which is the Wulgurukaba and the Bindal people, and the traditional owners of the waters where the data for this project will be collected. I pay my respects to elders past, present, and emerging, and recognize their roles as Australia's first scientists, first innovators, and custodians of the land. So here we go. This is a bit of an outline of what we're going to go through today. And then yeah, I'll just get right into it. So a little introduction. The giant shovelnose ray is a shark-like ray. It's a member of Glaucostegus, which is the giant guitar fishes. So they are classically misidentified as sharks due to their strong tail and then two prominent dorsal fins. However, unlike sharks and rays, they actually possess no means of hurting humans. So no stingers, no barbs, and pretty much their teeth won't hurt us. So they have a really large distribution around the coastlines of 18 different countries, spanning all the way from eastern Indian waters all the way over to western Pacific waters. And actually some of their healthiest populations are here on our coastlines in northern Queensland. So although we assume they occupy a mesopredatory role in their environment because they eat and are eaten, we actually don't really know much about the relationship with their environment or how they support it or structure it in any way. So another knowledge gap. So just a bit about their habitat and ecology. So their habitat and diet change with age. So the adults tend to be out in these deeper waters, like intertidal, deeper outshore water regions. And the young of the year, for example, they are in more intertidal coastal areas like sand flats, mud flats, mangroves, nearshore shallow environments. And so while we do have some preliminary information about what the adults are eating, we don't really have any about what the juveniles are eating, and especially in these areas, which is critical. And then with regards to growth, juveniles are classified as anything under 45 centimeters, but the adults are actually able to grow up to 4 meters in length. So with regards to their life history, they're very slow growing. We don't really know much about we don't have like a solid generation length, all we have is estimates, but it's assumed that at least, I think 15 is the average length of their generation, and the females don't reach sexual maturity until about 7 years. So you can imagine with a long generation and slow growth, they are incredibly vulnerable to population declines, and we've actually already started to see this, which is how they earned their listing as critically endangered globally by the IUCN. But it's not all doom and gloom. Here in Australia, they're actually locally abundant and classified as least concern by the IUCN. So I'll circle back to that point in just a second, but there's hope. So with regards to threats, I've narrowed it down to 4 main threats. The first of which is bycatch. So they're classically caught as bycatch in different environments for longline and bottom trawling mainly. And if you can see here in that top right corner, it's taken from La Paz, Mexico. It's after a shrimp trawling fishery had just discarded its catch back into the water. The bycatch from it. And so, yeah, that's pretty much just random juvenile shovelnose rays, which it's not great for, populations. And then we also have habitat degradation. So the destruction of, coastlines from development and runoff and pollution and all that stuff is really not great for their habitats or communities. So then we have the worst of all, which is data deficiency. This is just based off of what we know right now, but the reality is that we don't really have a full understanding of the threats that they face, and that is majorly impacting their conservation efforts. So then we have our research gap. So in a study in 2021 by Darcy and colleagues, they said that of the 611 stingray species that we know of today, 12.5% of those are classified as data deficient. And within that group of data deficient rays, we have the giant shovelnose rays and guitar sharks in general, which are some of the most endangered and vulnerable due to the lack of data and studies on them in general. So I have identified the lack of data on their feeding, ecology, and nursery environments in these intertidal areas, specifically the juveniles, as my main knowledge gap that I'll be focusing on. However, the family of Glaucostegus and just guitar fishes in general are kind of their own knowledge gap. But circling back to what I had mentioned before, we're actually in a really fortunate position because they're pretty much locally abundant in our backyard. So this is an amazing opportunity to get to study them. So this brings me to the aim of my research. I am primarily going to be looking at understanding the foraging ecology of giant shovelnose rays in these high density areas, primarily the juveniles. And then these are the research aims. So I will address my aims by describing their diet in these intertidal nurseries and, identifying their diet in these intertidal nurseries, as well as identifying potential differences in diet and feeding strategies between a few locations. So this is an example of what I mean by high density areas. So we already have a lot of data, thanks to ongoing research from PhD candidate Jalen Myers, of the diet of juvenile shovelnose rays over in Lucinda. And so I'm going to be looking at one or two different areas that have similar habitat features and densities of these rays. So right now I'm looking at Tulukia or maybe Kangala to do that. So this is just an overview of my methods. I'm going to be performing a gastric lavage, benthic invertebrate sampling, and lab analysis. So let's get into some details. To start off with, this is going to be a gastric lavage. Gastric lavage is a very common means of getting non-lethal diet samples, just for the background. So the rays will be caught using dip nets and save nets and then promptly placed into a tub with a mesh netting over it, like it has in this bottom corner, and some water for the ray to remain comfortable. Next, I will measure the ray, take a tissue biopsy, and then attach a dorsal tab to help with future study and recapture data. So to perform the lavage, pretty much the ray will be flipped onto its back, as it is in this one, and then a appropriate size tube will be placed down the mouth and into the base of the stomach, at which point the bilge pump will be turned on to an appropriate pressure and then gently flush out the stomach contents for a maximum of 10 seconds. After this, we'll release the ray to minimize any stress as much as possible, and then pick up all the little invertebrates that came out into the mesh tray and put them into a little diet tube. And then release the ray. Yeah, the ray's out there swimming around. So next we have benthic invertebrate sampling. I'll be employing two methods to do this. The first is an empty benthic sled, which is pretty much, I'll drag a weighted sled attached to a net that gets funneled down into a little sample tube across the sand flats. And then I'll also be looking at sediment cores, where I'll core the sediment, and then use a sieve to sift through it, and then pick out the little invertebrates and get those out to the lab for sampling. But both of these will be used to assess the availability and prey distribution among key microhabitats within those 1 to 2 larger study sites that I'm looking at. And then finally, we have lab analysis and ID. I will be pouring out all of these beautiful dye samples onto the tray, just as Jalen has here in this picture of her research, and using a benthic invertebrate ID book to identify what invertebrates were in that sample for the diet samples and the sediment samples. With regards to data analysis, I will be classifying all my organisms in my diet samples to the family level or higher, and then I'll be able to calculate a frequency of occurrence based off of presence and absence data of the specific species that have appeared in those specific samples. And then I'll use this to determine what prey items were actually dominant in those diets. Additionally, I will create a non-metric multidimensional scaling plot, which will help me visualize the level of similarity between different individual rays diets across the 2 different locations to see, well, 3, I guess. So for expected outcomes of this project, so I will produce a list of key prey items in the diets of juvenile shovelnose rays in these intertidal nursery areas, which has not been done before. And I will also create a description of the feeding ecology across different sites to see how that might vary, and put that into the literature, hopefully. So some challenges. Ray catching is quite literally a physical challenge, so I felt that was important to include that. It definitely takes a lot of manpower and proper planning, which brings me over to the weather and tides challenge of it all. So it's a lot of field work. You gotta go out and get the rays, but to do that, you need to have the right tides during the right hours, daylight hours preferably, and make sure weather, glare, wind, all the things are organized so that you can have the most effective field day. With regards to limitations to the study, I will only be sampling during daylight hours to avoid crocs and other risk factors like tripping. And then also that would potentially leave out any type of variation that happens throughout the day into the night. So just something to take note of. Also, obviously, this is kind of a short time frame. I have a year to complete this project, so I'm not going to be able to really take into account any type of seasonal variation in the diets across a longer time scale, but maybe one day. And then this brings me to my last slide, which is the dietary, why does it even matter? How does dietary analysis help us conserve an endangered species in any way? It seems pretty removed. But at the end of the day, dietary analyses are actually crucial in conservation biology because they provide specific insights into the feeding habits and nutritional needs of these rays. So by understanding what they eat and how they interact with their environment, we can develop targeted strategies to both protect and restore the habitats they belong to, ensure prey availability into the future, and then just ultimately create more effective conservation plans that are, diet specific to that species. So it's important to preserve these high value nursery areas because this is where the next generation is getting raised. So if we can get more information about what is there, that will go a long way for conservation. And so, yes, this knowledge will help us to sustain these species and maintain their ecological roles within their ecosystems, even if we don't know what those ecological roles are at the moment. Gives us time. So here's just a brief overview of what my timeline looks like. I've recently completed my animal ethics training course. I'm on to my literature review and research proposal at the moment, and we'll start planning some field trips in the next coming weeks. And yes, I'd like to say just a quick thank you to everybody on my team. Thank you to Dr. Anna Martins and Professor Marcus Sheaves, who may or may not be here, and of course to Jalen for really just you guys helping me put this whole project together. It's been amazing. So thank you guys.